All right, hello everybody. It's me, Dr. Kaz. I hope everyone's enjoying themselves. And as promised, I am going to do the part one portion of metabolism in the supplemental video for everyone. So let's get underway. First, this video is going to probably be a little bit of a review. If you have an interest in biochemistry, you might find some of this information pretty helpful and interesting. If not, I apologize. But we talked a little bit about, biochemistry in Chapter 2 when you took the Biology 210 component of this course. So now we're going to kind of follow up and go over some of the parts and concepts that we discussed before in those other chapters. So obviously metabolism, and we touched on this a little bit when we were talking about the organismal levels and whatnot. So metabolism is basically going to be, all right, these biological reactions that occur in living organisms here, all right? And so obviously, since we are discussing biochemical, we have to deal with both biology and chemistry. So we will discuss, all right, certain chemical reactions that are in the body. And we've done that throughout the chapters here, okay? But we're going to kind of do a broad overview here. All right, so as you know, and we just got done with chapter 26, which was basically, with digestion, several chemical reactions. We're starting with a big structure and breaking it down into its smaller parts and pieces. That's essentially what you do when you digest things. All right, so how we do that is we have to break down the bonds in between the molecules. And eventually we can get them small enough where we're going to be able to absorb whatever the components are going to be and use them either to make proteins or our own fats or even recycle or create carbohydrates, okay? But we need to get things small enough here, all right, when we were talking about digestion, okay? So when we talk about metabolism, all right, we're going to discuss how we're breaking down some bonds, okay, and then how we're actually going to build new bonds. So some are broken and some bonds will be made, depending on what type of metabolism that we're going to discuss, okay? So when we talk about certain reactions in the body, we do so by using what we call a chemical equation, okay? And it's basically like our recipe. We're going to set up our recipe, tell you what the ingredients are, and then tell you what's going to happen, right, to those ingredients and what's going to result. All right, so like when you're baking brownies, you're going to start off with eggs and batter and whatnot, put it in the oven for X amount of time on a certain temperature, and then you're going to wind up with brownies, okay? So in this case, when we're dealing with chemical equations, all right, we're going to have two items in a chemical equation, all right? Reactants and products, okay? So reactants are basically what we're going to start off with, and then the products are going to be what we're going to end up with, okay? So reactants are going to be what's present prior to the start of our chemical reaction, okay? And so when we're doing our chemical equation, we'll see them on the left side of the equation. And the products are what we're going to wind up with, all right? They're what's formed because of this reaction, whatever type of reaction we're going to discuss, and we'll see those on the right side of the equation. So the example here, A plus B, all right, gives us C. Okay, A and B are the reactants. All right, those two items are going to interact with one another, and then C is going to be the product here. Okay, and so an example of that would be when we're making hydrochloric acid that we see that the parietal cell in the digestive system is very helpful with. All right, so we have our hydrogen ion, okay, and then we're going to combine that hydrogen ion with chloride. Okay, and then our resulting product will be hydrochloric acid, all right, which, as you know, all right, is vital, quite important, okay, to the chemical digestion, all right, and even some of the mechanical digestion of certain products that enter into our body. Okay, we see how it helps with proteins, all right, and the fact that it will activate the pepsinogen into pepsin and help to digest proteins chemically. Okay, also helps to physically break down some larger structures. Okay, so we're going to see this, and again, this type of scenario here, when we're talking about chemical equations, is universal throughout the body, and we're going to see it in several different areas, all right, and in several different systems here. Okay, so you can see the arrow here is going to tell us which way we're going to go, because you can always reverse the arrow, all right, when we're talking about breaking down hydrochloric acid into hydrogen ion and then the chloride ion there, okay? So when we talk about balanced equations, we're basically saying that you've got equal parts on both sides of the reaction, okay? So meaning you have, all right, our products, or excuse me, our reactants, A and B, all right, we're going to wind up with equal parts of products, which would be C and D, okay, if we augmented that reaction in the previous slide here. All right? So when we talk about, all right, the classification of these chemical reactions, there's three criteria that we're going to use, and we're just going to discuss the first one here, changes in the chemical structure. All right, you're also going to see changes in the chemical energy, and then whether or not a reaction is irreversible or reversible. Like I said, we're only going to focus on the first one here, changes in chemical structure, because we're just going to be glossing over this. This is not a biochemistry course. This is just an anatomy physiology course. Okay? So when we're talking about classifying the changes in the chemical structure, there's two reactions that I really want to focus on: decomposition reactions and synthesis reactions. All right, we're really not going to talk about exchange reactions here. All right, so decomposition reactions and synthesis reactions. Now, if you think about it, you're somewhat familiar if you look at the terms here, all right, of these types of reactions. All right, if you were ever into, you know, when we talk about decomposition, all right, we're going to start with something like if you do composting, you throw a bunch of stuff in a pile in your backyard or in some box or in some drum, all right, these big leftover like eggshells, banana peels, whatever, and then over time it decomposes, you've heard that term, and it breaks down into smaller things. And that's essentially what decomposition reactions are going to do. You're going to start off with a big item and it breaks down into smaller items. All right, so the type of terminology that we use to describe decomposition reactions are catabolism. All right, you start with a large molecule, it gets broken down into smaller structures. All right, now with synthesis reactions, all right, just the term of synthesis, we're going to make something. All right, so in that type of situation, synthesis reactions were also known as anabolism. All right, and in synthesis reactions, what we're going to do is we're going to take a couple small items, all right, 2, 3, 4 smaller items. All right, we're going to combine them together, and we're going to wind up with a larger item, all right, or a structure here. All right, so let's start off with the decomposition reaction here. All right, so here you can see in our example, all right, we're starting off with a large structure here. For example, you could use a water molecule if you want, all right, or the example listed down here is table sugar, okay, sucrose. All right, and so in our body, we saw that the brush border enzymes do this, all right, in our digestive small intestine tract there, all right, how we have our brush border enzymes will break sucrose down into our monosaccharides of glucose and fructose, all right? So we'll see, for example, all right, hydrolysis. Now, hydro is going to include water, which means we're going to add water into this chemical equation, all right? And when sucrose is being broken down, all right, we're going to break up water, all right, into, all right, hydrogen and hydroxide, all right, and we are going to donate those elements to glucose and fructose here, okay? So when we hydrolyze, all right, sucrose, we break it down into our monosaccharides of glucose and fructose, okay? So this type of reaction, yes, it's a decomposition reaction, but we refer to it as, all right, a catabolic reaction, or the general term when we're just describing metabolism is catabolism, starting with something large, breaking it down into something small. So think of this as digestion, okay? The next one here is going to be our synthesis reaction, all right, otherwise known as anabolism, okay? So we're going to take two or more structures, combine them together, make one big, large structure, okay? So the example that we're going to use down here is a dehydration synthesis reaction. So we're going to take individual amino acids, 2 amino acids, okay, and we're going to form a bond between these 2 amino acids, a peptide bond. All right, and as a result of that formation of that peptide bond, all right, our byproduct for this reaction is water, okay? So hence dehydration, right? Because of this reaction, we're dehydrating, all right, the reactants, and one of our byproducts is going to be water, okay? So those 2 amino acids attach to one another through that peptide bond. So we started off with 2 amino acids and we wind up with a dipeptide. Okay, so we took 2 small items, combined them together, and now we're left with a larger structure. All right, that's anabolism. Okay, so when we start to see these synthesis reactions, all right, the actual formation, all right, this is the start of making proteins. All right, so these proteins are anabolic, all right, reactants, okay, that we are going to create, or products, I should say. Okay, all right, so that's just kind of a general broad overview. All right, when we're talking about classification of these chemical reactions, all right, and basing that classification on the items that are going to be included when we're discussing chemical structure. All right, so let's jump into now a topic that we talked about way back in chapter 2. All right, macromolecules. Okay, so remember the smallest item that we discussed way back in Bio 210, all right, were elements and molecules. Now, when we start to add molecules to one another, we're going to get macromolecules. And so macromolecules are going to include our carbohydrates, our lipids, our proteins, all right, nucleic acids. These are going to be some of the structures that are included in the macromolecules. All right, so now we're taking a step up, all right, from the smaller molecule size and elements, and we're going to jump into, all right, our macromolecules here. So the first one we're going to discuss are carbohydrates, and we briefly talked about this a little bit in digestion. All right, so when we're discussing carbohydrates, all right, we have 3 classes that we're going to discuss: monosaccharides, otherwise known as simple sugars, all right, disaccharides, and polysaccharides. So when we talk about, all right, bringing these carbohydrates in as dietary sources, meaning what we're putting into our mouths, okay, we have sugars, starch, and fiber, okay? So the starch is basically going, well, I'll get into it, but starch is just basically going to be glucose repeating monomers of one another, okay? So let's first start off with sugars, okay? That's going to be our first 2, both the monosaccharides and the disaccharides. All right, so you really should know this and really make it a point to memorize the monosaccharides here: glucose, fructose, and galactose. Okay, so when we are dealing with glucose here, all right, it's a hexo, it's a 6-carbon carbohydrate. All right, so when we're dealing with, all right, our monosaccharides, when we attach 2 monosaccharides together, we'll get a disaccharide. Obviously, you can just determine that from the name here, all right, or the title of these classifications of sugars, okay? So when we have 2 glucose monosaccharides attached to one another, we get maltose, okay? So that's G and G, glucose and glucose, all right? When we attach glucose to galactose, then we get lactose. All right, and then we get my favorites. All right, when we attach glucose to fructose, then we get sucrose. Okay, so glucose is at least one of the monosaccharides that's going to be involved, all right, in our disaccharides. Okay, so if it's 2 glucose molecules, it's maltose. All right, if it's glucose and galactose, it's lactose. And then if it's glucose and fructose, it's sucrose. Okay, so some of the other sugars that you're probably familiar with: dextrose, brown sugar, honey. Again, right around this time of year, we're seeing lots of people really enjoying honey, especially if you're dealing with allergies. Okay, apparently, you know, taking honey in, all right, from the local area here helps to decrease, all right, your allergies, due to the fact that if your allergies are propagated through pollen, all right, the honeybees use that pollen to help make their sugar, or their honey. And if you take in some of that honey, your body gets used to it and can help to be exposed to it, so your reaction with the allergies is less severe. All right, so staying with our carbohydrates, starch. We talked quite a bit about that in the digestive chapter there, when we were talking about it being what we refer to as a complex carbohydrate. Okay, And so what we're going to see with starch is, like I said before, it is a polysaccharide, all right, which is going to be multiple monosaccharides linked to one another. But in this case, it's a polysaccharide polymer of glucose. What does that mean? All right, that means it's a big long chain of glucose monomers just strung along one another. Okay, and you'll see it, and we're going to talk about the different types here, all right, both in plant and animal, all right, and what we call it. Okay, but starch, just think of it as it's a polysaccharide, and it's just repeating glucose molecules. All right, we're going to find it within certain foods. All right, tubers, grains, beans, potatoes, quite commonly, you know, the white portion of the internal portion of the potatoes is going to be pretty much the starch. Okay, then fiber. All right, that's the part that we can't really digest. All right, comes from plants mainly, but so, you know, we'll get fiber from animals also. All right, our problem is we cannot get it down. We can mechanically start to break down fiber and digest it, but it's the chemical issue that we have problems with. And when it comes to plants, it's because of the cell wall in the plants. All right, remember, plant cells have a cell wall. Animal cells do not. Animal cells will have the plasma membrane. Okay, so we can't chemically digest the fiber. We can't absorb it into the GI tract. All right, so it's going to act as filler and just kind of get pushed through the GI tract, which is good because, believe it or not, it does help with digestion. All right, and it's very important. It's always encouraged in diabetics, especially because they already have enough issues with insulin production and insulin sensitivity, where fiber will actually help to promote, all right, absorption of this material, all right, especially during the digestive processes here. All right, so let's talk about everybody's favorite simple sugar, glucose. All right, yes, it is the most common monosaccharide, and your body loves it. All right, it is the primary nutrient, and you'll see it quite often. All right, in which we have situations, we even have a process called gluconeogenesis, in which your body will use non-carbohydrate sources to make glucose. That's how much it loves it. It'll convert other macromolecules, all right, and other substances into glucose. All right. So your body's always trying to maintain proper what we call blood sugar levels. All right, remember, the brain is the primary organ for pretty much everything in trying to sustain, all right, homeostasis. So it's really important. Whatever the brain wants, the brain gets, essentially. Okay, so, and it's one of the amazing things is that your central nervous system, okay, your brain does not need insulin to absorb glucose. Most tissues, all right, muscles need tissues, liver cells, need tissues. Muscle cells need insulin. Your liver cells need insulin. All right, in order to absorb, okay, glucose. Well, guess what? Your brain and a couple other tissues do not, which is kind of nice. You know, it's really important, all right, in regards to that. But at the same time, you know, it's again what the brain wants. The brain is going to get. So keep that in mind. I'm trying to remember a couple other structures that don't need, red blood cells. And I take that back. The hepatocytes actually do, all right, don't need insulin, okay? So pretty much brain, red blood cells, and the liver cells, the hepatocytes don't, which is important. So the term here that you see on the slide here, glycogen, okay, that's the storage form of our glucose. And glycogen is going to be, all right, a chain of monomers of glucose, all right, of those monosaccharides strung together, okay? And so the cells that are going to be primarily responsible for that are going to be the liver, obviously, and skeletal muscle. So when you're in what we call the fed state, FED, all right, and you have an abundance of energy, all right, from the food that you just ate and absorbed, okay, well, the leftover excess glucose will be stored as glycogen, all right, in the liver and in the skeletal muscle to be used at a later time. Now, when that later time comes, all right, the liver is going to hydrolyze. Look at that term there. All right, what's hydrolyze? Hydrolysis is a catabolic reaction. All right, so the liver is going to break down that glycogen. It's literally just going to start snapping off, all right, glucose monomers off of the glycogen molecule. Okay, and then it'll use that glucose as it sees fit. That process is called glycogenolysis. Okay, glycogen and then lysine. We're cutting glycogen, all right, pieces off the glycogen. Okay, and now, all right, when we're in a situation where we've run out of glucose and yet we still have tissues in the body like, hey, I want some glucose, go find me some. All right, your liver says, I got one better. I'll go make you some. Okay, and it's going to recruit non-carbohydrate sources. Okay, lactate is an example. All right, amino acids is another example. We'll talk about that later on in this chapter. All right, we're going to find these non-carbohydrate sources and make glucose from it. And that's called gluconeogenesis. All right, gluco referring to glucose, neo meaning new, genesis is making. So we're going to make new glucose. All right, that's going to occur predominantly in the liver. All right, so here you can see on this slide here how we have, here's our glycogen molecule. All right, we've drawn it nicely with these glucose molecules. All right, that they're in the shape of a hexagon. All right, so we refer to them as hexose monosaccharides. All right, 6-carbon ring here. And what we're doing is we're just going to start chopping them off. Chop one, chop the next one off. All right, as we chop them off, all right, we're going to start to break down the glucose molecule. All right, hopefully, all right, the end result will be to get that glucose molecule and all its substrates into the mitochondria for the Krebs cycle, so we can get our ATP, our 32 ATP. Okay, but if not, all right, if we can't get it in there, then we'll settle for 2 ATP. Okay, which is what we'll talk about later on. All right, so a couple other types of carbohydrates. All right. We talked about galactose and fructose. Those are the other isomers. And if you don't remember what an isomer is going to be a molecule, all right, that, and I'll use the example here of glucose, all right. Galactose and fructose are isomers of glucose. They have the exact same number of carbon molecules, hydrogen molecules, and oxygen molecules. They're just arranged differently. And so that's what an isomer is. All right, it's going to have the same recipe, but it's just going to look different. All right. Then we also have our 5-carbon monosaccharides, our pentose sugars. These are important because if you all remember, all right, when we were talking about DNA, RNA, ribose, all right, and deoxyribose, those will make the backbones, all right, of our DNA. Those are called pentose sugars. Okay. Disaccharides, we've talked about this already. It's our 2 simple sugars that are bonded together. All right, so we have our table sugar, which is sucrose, and that is going to be glucose and fructose. And then we have our milk sugar, lactose, all right, which is made up of glucose and galactose. And then finally our malt sugar, which is maltose, which is just 2 glucose molecules jammed together. Okay? All right. And finally, not to forget about our polysaccharides, all right? This is when you have 3 or more sugars, all right, strung along, all right? So I talked about glycogen. That's what we see in animals, all right? And then our starch and cellulose, all right? Didn't talk about the cellulose so much. The cellulose is pretty much the fiber, okay? Starch is going to be, all right, what we discussed, all right, that's getting broken down like potatoes, okay? Certain tubers and whatnot. But it's the cellulose is pretty much going to be the fiber, which is going to be the non-digestible material, which is going to pretty much give us that filler. All right, so if you want to look at some pictures here, okay, just to kind of commit to memory, you can see that all the different monosaccharides here. All right, so here are the 2 isomers for glucose, galactose, and fructose. All right, and then you also have your 5-carbon sugars here, all right, pentose and deoxyribose. And these will be involved in RNA and DNA. Okay, and then you can see your nice disaccharides. But notice each disaccharide here has a glucose molecule. here for sucrose, here for lactose, and then obviously at least one glucose, but here in maltose, it's just 2 glucose, okay? So keep in mind, I mean, the preferred form of energy in our body, all right, is glucose, but it is not essential. And it's not essential because guess what? We can make our own glucose thanks to the liver, okay, through gluconeogenesis. So if we're not getting it in our diet, no problem. You know, it's just the liver's got to take one extra step and start the process of gluconeogenesis. But your body, all right, a lot of the tissues prefer, all right, glucose, all right, for its primary energy capacity there, okay? And then lastly, we're talking about fiber here. Yes, I talked about adding bulk, all right? It does help to stimulate peristalsis, and if you recall, peristalsis is that process of moving chyme and food product through your GI tract from proximal to distal, okay? And it actually does help to lower the cholesterol. So there's another reason to make sure that your fiber levels are reasonable. You'd want to take in anywhere from 32 to 35, all right, grams of fiber a day, okay? Grams. Notice I didn't say milligrams, grams. Okay, 32 to 35 grams of fiber a day. Okay, why? Keeps you regular. No, it helps to lower the cholesterol, and especially if you're one of those folks that could be borderline diabetic, or even if you have diabetes. All right, it helps, all right, with the absorption of sugars. All right, let's jump into our second macromolecule, and that is going to be lipids. Okay, fats. Think of lipids as fats, but most importantly, they're fatty, water-insoluble molecules. Okay, which means if you put a fat into water, it will not dissolve into that water. Okay, it will not separate. Okay, so hence it is water-insoluble. Right? The functions here listed for our lipids are stored energy, cellular membrane components, and hormones. We'll check it out. Stored energy, triglycerides, okay? Cellular membrane component, phospholipids. Remember the phospholipid bilayer, okay? And then finally, hormones, all right, that's going to be steroids, and then our locally acting hormones, the acostanoids, okay? So now you can see, and some of this, you know, first when I throw this slide up, this is the exact replica of the same slide that I put up in chapter 2. We're talking about biochemistry. Okay, so you don't even think about it, plus you're starting off in the course. But now, all right, as we move through this course and you've seen, all right, a lot of the functions, especially after chapter 17, when we talked about the endocrine system, all right, number 3 and 4 should absolutely make sense. Number 2 should definitely make sense after you took chapter 4, which is when we learned about the cell. And then triglycerides, we'll talk more about it in this lecture, but also you saw a little bit of it in digestion. We were talking about the breakdown of triglycerides, okay, and what molecules make up the triglycerides. Okay, so speaking of triglycerides, let's talk about triglycerides. Energy, energy, energy. So these molecules are great for what we describe as long-term energy storage, okay? So first of all right, we reviewed it in chapter 26 in digestion, that a triglyceride is a glycerol group with 3 fatty acids attached, okay? And those fatty acids can vary in length. They don't have to all be the same length. And I mentioned this before. They're what we call short chain fatty acids, medium chain fatty acids, and long chain fatty acids, right? So essentially a fatty acid is going to be, I'll show you a picture here just so you can kind of get an idea. Let me just zoom in here. Okay, so here's our picture of our triglyceride. Here's our glycerol group here with the 3 fatty acids attached. Now you can see, all right, they're not all the same length. All right, but fatty acids are made up of, if you can see, carbons and hydrogen molecules attached. All right, but here's what I want you to think of. Each of these bonds here, think of that as energy, okay? So when your body goes through the process of breaking these bonds, it's going to get energy from the bonds. So the longer the chain is, the more energy you can get from them. Okay? Keep that in mind, please. Okay, so our fatty acids are going to have a glycerol group. Excuse me. Our triglycerides are going to have a glycerol group and 3 fatty acids, all right, attached to them. Okay? So I mentioned that they can vary in length, but I didn't talk about the number of double bonds. Okay, so if we don't have any double bonds, all right, and this is where you probably heard these terms before, all right, saturated versus unsaturated versus polyunsaturated, all right, and then you've heard the term trans fatty acids. All right, we'll talk about that. Okay, but if we refer to a fatty acid as saturated, all right, that means there's no double bonds, none. Okay, they lack a double bond. If it's unsaturated, that means there's at least one double bond somewhere in that fatty acid. And then if it's polyunsaturated, it's 2 or more. Okay, so unsaturated, no double bonds. So that picture that I was showing you here, all right, this fatty acid, all right, right here, is a saturated fatty acid. There are no double bonds in there. Okay, now the one above it, all right, since it only has one, okay, that's an unsaturated fatty acid. And the one below only has one also, okay? But if we saw 2 or more, right, then it would be polyunsaturated, okay? All right, so a review of what we talked about, okay, in 210, all right, when we're in a situation of the fed state versus the starvation state, or what we call the fasting state, okay, your body, all right, depending on what type of metabolism you're engaging in, and what I mean by that is it aerobic or anaerobic, all right, what's available, okay, but your body ideally would like to use, all right, triglycerides, okay, because you get a lot of energy from a triglyceride molecule. Okay, but, all right, depending on what state you're in, if it's the fed or fasting state, will determine what type of metabolism occurs. Okay, so for example, you've just eaten a nice meal, all right, all the tissues are satisfied, they're full. Okay, but your body's like, well, I don't want to get rid of, all right, these lipids. All right, I want to keep some of this material. We'll discuss this process here later on. We'll get into the details of this. All right, so when we have an excess, when you're in the fed state, okay, your body will engage in lipogenesis. Okay, this is when it's going to make triglycerides. Okay, and it'll take those triglycerides, it'll package them up, and eventually the end result will be adipose tissue. We're going to store it in adipose tissue, all right, as lipid droplets. Okay, now you're in a situation where you haven't eaten in days, all right, and your body needs some energy, okay? So you are in the fasting state. Then your body will engage in lipolysis, all right, in which you will start to break down triglycerides, okay, that are stored in the adipose tissue, all right, so you can get energy from that. And again, later on in this chapter, or chapters, I should say, we'll discuss that. Okay? So you have lipogenesis, the formation of triglycerides, then you have lipolysis, which is the breakdown of triglycerides. Okay, so what you're seeing here, all right, is basically showing us, all right, here's our triglyceride. All right, each fatty acid chain is attached to the glycerol molecule here. All right, so when we want to break, all right, this molecule down so we can start to digest everything, we're going to engage in lipolysis. So now we're going to disengage, all right, the fatty acids from the glycerol group. All right, as that occurs, all right, you're going to see, all right, the formation of water occurring. Okay. All right, and then when we have plenty of food, because you know from chapter 26, all right, when you eat fat, all right, we start to break it down in the mouth. It ceases temporarily in the stomach, only to resume again in the small intestine. All right. So the salivary amylase is going, or lipase, excuse me, is going to start to break down triglycerides into a diglyceride and one free fatty acid. And then when we get into the small intestine, then we're going to further break that down into a monoglyceride and two free fatty acids. Okay? And we'll see as we're starting to disengage this, we can start that process, all right, is going to be, that's our lipolysis when we start to break it down. Okay? Oh, I lost my train of thought. So we start to absorb all of those molecules into the intestinal cells there, all right, where in the intestinal cells we'll repackage them, all right, through the process of lipogenesis. And then we'll send those, remember we talked about the chylomicrons, okay, in which the chylomicrons, all right, which are proteins that we just wrap these proteins around the fatty, or excuse me, around the triglycerides, so we can export them into the bloodstream so they can go to the liver, and the liver can decide what to do with them, which we'll talk about later on. All right. Okay, so that is called, all right, lipogenesis there. Cool stuff, cool stuff. We'll get into more details about it. All right, phospholipids, the next group of lipids, all right, are the phospholipids. Now notice this first term on this slide here calls them amphipathic. That means, all right, one part of this molecule is going to have a polar portion to it, and another part of the molecule is going to be nonpolar. So a phospholipid is similar, okay, similar to a triglyceride, except it only has two tails, okay, instead of three fatty acids hanging off, okay? So the head is the phosphate group, and that's the polar portion. Easy to remember. Phosphate begins with a P, polar begins with a P, all right? And then we've got our fatty acid tails here, all right? Those are going to be nonpolar, all right? If it's nonpolar, we refer to the tails as hydrophobic. Hydrophobic meaning water-fearing. They do not like to be with water. They don't mix. Hydrophilic, that's the water-loving portion, all right? That's going to be that phosphate end there, all right? And that is going to be water-loving, hydrophilic, okay? You know what else is hydrophobic? All right, rabid dogs. They do not like water. That's true, okay? That's true. Okay. So those are phospholipids. Where do we find them? Cell membranes, okay? Cell membranes. All right, and so we have cells everywhere, so that's why phospholipids make up a large number of lipid composition in our body, okay? All right. The next class is going to be the steroids. Now, we reviewed this and we talked about this in chapter 17. All right, our steroids are what we call hydrocarbons. Now, if you don't remember what a hydrocarbon is, dissect the word every time if you're not sure. All right, it is going to be, all right, a molecule that has just hydrogen and carbons in the molecular formula here. Okay, so these hydrocarbons are arranged in a 4-carbon ring. Okay, the first three rings have 6 carbon atoms in it. The last one has 5 carbons in it. Okay, important thing is when we talk about steroids, all right, now we notice, you know, steroids are going to be important when we're talking about, all right, certain types of hormones, the sex hormones, all right, for males and females. Okay, but these steroids will differ, all right, because attached to their 4-carbon rings, they'll have these what we call side chains. And whatever that side chain is going to determine, all right, what function that molecule has. Okay? So when we're discussing cholesterol, all right, its side chain is going to differ from bile salts, but both are considered to be steroids. Bile salts and cholesterol will differ from actual hormones or steroid hormones, all right? So keep that in mind, all right? We know the importance of cholesterol, one, all right, because we're going to use cholesterol in a lot of different things, all right? It's going to be needed for hormones and bile salts, but also plasma membranes. Cholesterol allows the plasma membrane, especially when we literally turn up the heat, all right, it allows the plasma membrane to be more resilient, all right, in temperature extremes, both in hot and cold temperatures. And the more cholesterol you have in the plasma membrane, this is why fat, believe it or not, or cholesterol is important in your diet, okay? This plays a role, okay, in your cell's ability to withstand these temperature extremes, okay? And then also, we need it for certain steroid synthesis. Bile salts also, all right? Oh, there's a drug out there, and I cannot remember the name of the drug. Remember we talked about that bile salts get recycled? All right, pretty much 90% of the bile salts get recycled in your body, okay? And you get rid of 10% through your feces. Well, there's a drug, all right, that folks that they give to folks with high cholesterol, in which it decreases, all right, the bile salt recycling rate, and so you'll excrete more bile salts. So if you're excreting more bile salts, the liver has to make up for those lost bile salts. What's it do? It's going to make more of those bile salts, and it needs cholesterol as an ingredient. So it'll where's it going to get the cholesterol from? Your blood, all right? Because you just absorbed it from that McDonald's meal that you just ate that was loaded with cholesterol. So this bile salt drug is going to force your liver to get cholesterol where it can, and it'll get it from your blood, thereby decreasing, all right, decreasing the amount of cholesterol, all right, that is in circulation. And then finally, our last classification, all right, or group of lipids is the acostanoids. Okay, so I discussed them. I said that those are the locally acting hormones, all right, but they are made from, you need to know this, all right, arachidonic acid. Okay, it's found in the plasma membrane. It all starts with arachidonic acid. Okay, so if you look at any type of enzymatic cascade, when you're talking about, Google it right now, all right, and pull up the inflammatory pathway. If you look at the inflammatory pathway, all right, the first thing that should be at the top of that enzymatic cascade should be, all right, arachidonic acid. If you're not quite sure what I mean by enzymatic pathway, basically, all right, when we produce, all right, these 4 classes of acostanoids down here, okay, we all have to start with arachidonic acid. And there's certain enzymes that are present in your body, in the cells and whatnot, that are going to promote certain chemical reactions, okay? And so these enzymes will go, you'll start with product A, it'll undergo a chemical reaction, then you'll get product B. Then you'll have a different enzyme that's going to act on product B to give you product C. That's our enzymatic cascade here, all right? So we all start off with arachidonic acid. And at certain points through this whole cascade of events, you'll get these 4 classes, all right, of acostanoids: prostacyclins, prostaglandins, thromboxanes, and leukotrienes. We've talked about a few of these when we were talking about the inflammatory effect when you damage a blood vessel. We talked about the immune response there and what happens. All right. So we're going to see that these acostanoids are going to play a big role in the inflammatory response. Not only that, nervous system communication. Okay. So we're going to see how these acostanoids, right, can have certain effects on your nerves, pain production, for example, pleasure reaction, okay, vasoconstriction, bronchoconstriction. All right, all these acostanoids can affect those tissues through how it interacts with the nervous system. Okay. So let me just jump into last slide here for this video, and then actually that's a lie. That's a lie. I got one more after this, but let me talk to you about the fatty acids here, about saturated, unsaturated, and trans fats. Okay. So keep in mind, all right, what did we say? If something is saturated, that means that it does not have, all right, a double bond in it. Okay, so most animal fats are going to be saturated. All right, most animal fats. All right, something you want to understand when we talk about lipids. If a lipid is at room temperature, pretty much it's going to be solid. Okay, and so usually we refer to that as a fat. All right, if we're discussing lipids and it's liquid at room temperature, then we refer to that as an oil. Okay, same thing though. Okay, it's just the physical state that it's in. Okay, so when we talk about most vegetable fats, those are going to be unsaturated, so they're going to have at least one double bond. Right, and so most of those will be liquid at room temperature. Okay, yes, these are going to be the healthier alternative. Okay, but we can convert, all right, these unsaturated fats into saturated fats, and that process is called hydrogenation. I'm not going to get into the details of this, but during that hydrogenation process, all right, if we don't completely undergo, all right, that hydrogenation process from unsaturated to saturated, we wind up with our trans-saturated fats or trans fats. All right, that's the bad stuff. That's the stuff, all right, that's been outlawed literally in New York City. Okay, no trans fats, can't have it. Why? Because it makes you more at risk for certain health issues. All right, like a CVA, a stroke, all right, heart attack, all right, atherosclerosis. Okay, these are bad. Trans fats, no bueno, you don't like it. So what's a trans fat? It's a partially hydrogenated fat. We didn't complete the process there, right? By design, all right? It wasn't by accident. I mean, well, initially, all right, but it was by design. Okay, and then finally, our polyunsaturated fatty acids, 2 or more double bonds, all right? We're going to see these are going to be liquid. So these are the ones that well, when I say we like, I'm not saying that they're the best, because I got to tell you, I've done a lot of research into the different alternative sources, all right? What's a good oil? What's a bad oil? All right? Some have, you know, there's a lot that have benefit and there's a lot that don't, okay? So you have to do your own research, and it really comes down to risk versus reward. All right, olive oil for a while there was getting a lot of press and saying how good it was. You know, the problem is if you heat olive oil for a long period of time, you start to break down the bonds, and then all the beneficial aspects to olive oil you're just thrown out the window. Okay, so keep in mind, I'm not going to discuss what's going to be, you know, healthy for you and what's not going to be healthy for you. You'll find along the way. But I mean, there are going to be pros and cons for most of these things. That's why I always just go with butter. You know, it's like eggs. Eggs are good for you, and then a couple years later, eggs are bad for you. And then a couple years later after that, eggs are good for you. Now they're bad for you. Regardless, I like butter. Butter tastes good. It's just like anything. Moderation. That's how I believe, you know. Is it bad that I go through 2 sticks of butter a day? Maybe, all right? But no, just kidding, I don't. But if I would if I could. But again, it all comes down to moderation. And then finally, our cholesterol here, okay? Big role in plasma membrane. I already talked to you about that, you know, and how it helps with the membrane resiliency here. But we're going to see, all right, is going to be a precursor, all right, which is going to be something that we're going to start off with, all right, to make steroid hormones, bile salts, and we didn't really discuss it, but also vitamin D. All right, vitamin D is crucial. It's very important. We're finding all sorts of things that are important for vitamin D, but we discussed in chapter 6 when we talked about the skin. All right, vitamin D helps with the absorption of calcium from the small intestine. All right, we're also seeing how it helps. High levels of vitamin D help to decrease, all right, certain types of leukemia. We're seeing that it also helps with the immune system. All right. And there was a video out there, an article I read, came out of France, in which they're seeing, and it's not a huge number, but it's still significant, 17% of people, all right, they've noticed in people with low amounts of vitamin D, okay, that 17% of those people, all right, are more prone to get the COVID virus or infection. All right, or you can say it the other way around. Folks that have enough vitamin D, all right, are 70%, all right, less likely, all right, are able to combat, all right, coronavirus infection. Okay. All right. So that's going to be it for this video here. All right, that's the first part of this discussion. We'll get into we'll talk about how we make cholesterol and whatnot, because we're going to start to get into the interesting. Not that this other stuff wasn't interesting, but the stuff that I like to talk about, the biochemistry. We're going to see what happens, all right, to everything that we discussed that in chapter 26. All right, you've broken this stuff down. You've absorbed it into your blood. Now what's going to happen? All right, what are your tissues going to use these macromolecules for? All right, so we'll talk about cholesterol synthesis in class, all right, in our next video. I hope this video was helpful, and I hope everyone's doing well, and I will talk to you soon. This is Dr. Kaz signing off.